Hey there, everybody. We'll go ahead and let folks start coming into our Zoom room here. Welcome, welcome. We are so excited to go over this webinar with you all. As people are coming in, I'd love to direct you to the chat box at the bottom of the Zoom window here. It should just look like a little speech bubble. And if you click that speech bubble and set your chat to everyone so we can all read your chats, go ahead and let me know let's see, what's a fun question? What was the last movie you saw in theaters? Go ahead and drop that in the chat and just make sure it's set to everyone. We will be answering your questions throughout the webinar, so it's helpful when your chat box is set to everyone. That just means that when we answer that question, people can refer back to the chat and say, "Oh, that's why he's talking about that." So, let me know what that last movie you saw was. Yeah, Candace, I totally get that. I'm kind of ooh, Oppenheimer, very cool. Yeah, I don't often see movies in theater, but when I do, it's one of those special ones. So we've got a few Oppenheimers. Love that. Love the history there. What's everyone else watching? Oh,
Dune
Dune
2.
2. We were just talking about that. Did you like it? What else have you all watched? I'm trying to remember. Honestly, I think Barbie might have been the last movie I saw in theaters, which
Barbie was
I
good.
loved. Yeah.
Barbie was excellent.
Oh, and it looks like Dune 2 got a big thumbs up from our educator friend in the comments and Oppenheimer from other folks. I love that. Oh, cool. Somebody did start of the year required videos. They do it at their local theater, which is really cool. All right, well, to make the most of our time together, thank you all for kind of interacting with us in the chat there. I did have some quick housekeeping items that I wanted to go over before we move into the rest of the presentation. And then if we could go to the next slide. Oh, perfect. All right, well, a recording of today's webinar will be live on both our website and our YouTube channel in about 3 to 5 business days after today's webinar. Keep an eye out on your inbox because you will receive an email with a recording link as well as a folder with additional resources and a certificate of attendance. As soon as I'm done talking here, I'm going to drop that in the chat, so keep an eye out for that. You can also message me on here and I'll send that to you again. And as I was saying, we'd love to answer your questions during today's session, so please feel free to use that chat section in Zoom to ask your questions. Just make sure that you have your chat box set to everyone so we can all read it. We will be monitoring that chat. If we happen to not get to your question, please reach out to us. You can email us at biology@vernier.com. And now that I'm kind of done talking, I am going to launch a poll just so we can see what you all are looking to get out of this webinar. And I will stop talking and get started. Thanks all.
Yeah, so go ahead and answer the pre-webinar questions for us. It's really helpful if you do that, because then we can focus what we're going to talk about in this webinar. Make sure that we focus that, switch it around if we need to. Just gives us a little bit more information so we can focus the webinar. In case we thought we were going to go in one direction, we can go in the other one. Great. Okay, that's exactly I think what Damon and I thought this would be. We have a nice mix of K-12 and college and other, but this is one of those webinars where we thought it would probably be more about the experiment than learning about Vernier sensors. And that's awesome, because that's why Damon is here, because this is a Bio-Rad webinar with Vernier, and it's a Bio-Rad kit and lab. So that's great. So we're on board there. So just to introduce the agenda, I just wanted to say that what we're going to talk about really is you're going to meet the presenters, which is Damon and I. Will introduce ourselves here in a minute. Then we're just going to do a basic introduction to glycolysis and fermentation. That's just to get everybody up to speed. In case you haven't reviewed glycolysis and fermentation, don't worry. We're not going to go through all of the steps and all the little pieces. It's going to be more like, hey, this is how this is related to glycolysis and fermentation. Don't forget, remember this, etc., and give you some good resources. And then we'll have an introduction to the biofuels enzyme kit from Bio-Rad. And then there'll be an introduction to the Vernier spectrometers, software, and lab books that are used with this kit. And don't forget, questions are welcome at any time. This presentation does contain a lot of links and online resources that should be useful later. So, and you'll have access to that as well. All right, so my name is John Melville. I have a bachelor's degree in biology and psychology from Sonoma State University. I got my PhD in zoology back at Oregon State University. I did a postdoc at the University of Oregon. And then I became an assistant professor of biology at a little place called Wartburg College, but I also was an adjunct at Lane Community College. And my big thing that I really liked teaching was non-majors courses in biology and neuroscience. I also taught marine biology a little bit, but I really liked teaching non-majors just as much as majors. And then my big emphasis was on what people call, I guess, active learning. And then I had a real emphasis on real-time data collection and then doing inquiry. So I wanted my students to actually design their own experiments and investigate things. And then another thing that I had a lot of experience at when I was at Wartburg is I helped train a lot of teachers that were going into the field. So I was really good friends with the one person that was training a lot of future educators. So I would hang out with them, show them stuff, etc. So that's me, and I'll let Damon introduce himself.
Hey everybody, my name is Damon Tighe. So I work for Bio-Rad Laboratories as a curriculum and training specialist. I cover kind of the Western United States, but also help out across the US on things like this kit where I'm probably the subject expert in the group. So I have a biology degree and a chemistry degree from St. Mary's College of California, which is a small liberal arts school in the San Francisco Bay Area. I went from there to go teach high school at Portland, Oregon, at basically a first-year school, probably the best way to get immediate gray hairs in your 20s, but also one of the most rewarding things you can do as well. My mom was an educator, and I remember her telling me in high school, don't ever go into education. And of course, being a son, that's the first thing you do is like, how can I defy my parents and go do that? Really rewarding, but I wanted to get back into bench science. And so I was looking at doing a PhD, but wanted to get some more hands-on experience. So I ended up moving down to the San Francisco Bay Area and got involved on the Human Genome Project. So at the Human Genome Project, I realized that going to grad school would be like going back in time 5 to 10 years. So I just never left. So I ended up being at the National Labs for about 10 years, working on large genomic projects, first the human genome, then working on single cell microbial sequencing, because you got to remember, the diversity of life on this planet is microbes, and we can't see most of them because we can't culture them. And so sequencing is the only way to see them. Within this, I was doing a lot of sequencing actually for biofuels to try and find these organisms that are good at breaking down cellulose, because we are a national lab that was being funded by the Department of Energy at the time. And then after those 10 years, I got to a point where I realized that one of the biggest obstacles that was taking place in our laboratory was finding good new employees, and that some of the ones coming in that had 4-year degrees were missing some critical skills. And so Bio-Rad, I had helped in my spare time build a DNA sequencing kit for classrooms, and they had this position open up, and I went, you know what, I think this would be a cool combination of my 2 careers, because I could help get more hands-on education going on into classrooms across the country. And so that's where I'm at right now.
All right. So I just want to say, Damon is the real expert on a lot of this. So I do know a little bit about fermentation and cellular respiration, but he really is the expert. But what I wanted to do is just remind everybody, I promise we'll go through this really quick, but just to get everybody on the same page is, when we're talking about biofuels and things like that, a lot of what we're going to come back to is the process of, well, really fermentation, making ethanol. But even before that, remember, we like to teach a lot about cellular respiration, and that involves glucose, or C6H12O6, as we call it, right? And then don't worry, this formula has not been balanced. That's okay, but this is, the formula for cellular respiration. Glucose plus oxygen produces, water and CO2. There's lots of other steps that are involved there, right? Like there's lots of other steps, but that's the formula that we like to write down. And then there's another process that we like to say isn't as efficient. It doesn't produce as much energy, but it's still really, really important, especially for lots of other organisms, and that's fermentation. And it produces this molecule right here, ethanol. But once again, glucose can be turned into ethanol and CO2, so it's also another form of respiration, fermentation. But I wanted to back up a little bit and just remind everybody that what we're really talking about are carbohydrates. And what are carbohydrates? Like if you haven't thought of carbohydrates before, or we should just really think about them just for a brief moment so you understand where we're going with all of this. And so what I wanted to do is there's some really amazing free resources out on the web that will go through all of basic carbohydrates if you're interested in it. There's the Khan Academy. Khan Academy actually took a lot of their stuff from OpenStax, which is a great resource. And then there's even a website where you can look at the molecular models in 3D. So I put these all here. But for example, if we just go to OpenStax, thank you, OpenStax, I love you. There's a great little, this is their biology, basically biology AP book. There's a great reference to what carbohydrates are and their molecular structure. And really, in a lot of cases, when we're talking about glucose, we're talking about a very specific monosaccharide. So it's like a monomer, right? So, and that's glucose here. There's glucose to hexose. There are 5 carbon sugars, 3 carbon sugars, and others. That's all related to the name, but we're going to be talking a lot about glucose. And remember that these all have slightly different structures based on where the oxygen group is. You can see over here that fructose is a 1, 2, 3, 4, 5. It's also a 6 carbon sugar, but it has a slightly different structure. And what's really interesting is they can also form ring structures. So this is alpha glucose and beta glucose, so they can turn around and form ring structures. And so and here's also ribose, and that will actually form fructose. You can see that's another type of sugar. So some of these monosaccharides actually have what I used to tell my students is different shapes. And it's going to be important what those shapes are for the enzymes to be able to break them down. These monomers can then form disaccharides. So like glucose and fructose can form together to form the disaccharide sucrose, table sugar. If 2 glucose molecules were to come together, that would form maltose. All right. And then there's other things, like here's lactose, which is galactose with a glucose. And then there are polysaccharides like amylose. This is a big, long chain of glucose molecules, starch, that can then be broken down into individual glucose molecules. And then cellulose is slightly different. So if you have any questions on that, just remember, this is a great resource that you can review. And then let's see. Great. Okay, that's been unlocked. What I wanted to just remind you is, remember, we spend a lot of time teaching about glycolysis, the citric acid cycle, and the electron transport chain. We consider all of that to be cellular respiration. But don't forget that there's fermentation, which is also really important. And the first steps in glycolysis are very similar for both. And Damon, if I'm saying anything wrong, remember to just correct me. So
Yep, I'll jump in there.
Great. So what I just wanted to review is in the process of glycolysis, what we like to say is, right, glucose gets turned into glucose 6-phosphate, and then fructose 6-phosphate, and then there's a bunch of steps, but the end product is pyruvate. Pyruvate can then go off into the citric acid cycle, or it can go through fermentation and become ethanol. Now, what's really interesting about this is there's lots of other sugars or carbohydrates that can go into this process. So glucose is a monosaccharide. It's pretty straightforward. It just follows through this process kind of in a linear fashion. Fructose can enter here. So fructose can just be turned into fructose 6-phosphate and then be turned into ethanol. So if you're going to make alcohol from like fruit sugars or fruit juice, that's that process. What about disaccharides? Remember, disaccharides are 2 monosaccharides put together. Well, typically you have to break down those disaccharides into their monosaccharides. But maltose, malt sugar, is really just 2 glucose molecules. So you just break a maltose in half, break it into 2 parts, and you have 2 glucose, which can then be used to create ethanol. Sucrose, remember we just saw sucrose is actually a glucose molecule and a fructose molecule put together. So sucrase is an enzyme that will break those in 2, and then those 2 molecules can then go into this process. Okay, so what about polysaccharides? Polysaccharides are really interesting because they're big, long chains of these monomers all put together. Amylose, which is also, you can think of it as just being starch, is just a big, long helical chain of glucose molecules end over end. And an enzyme can come along and snip those, and then they can just be put into this entire process. So if you could break starch down into its individual glucose molecules, you can create a whole bunch of ethanol. Of course, the next big thing there is, wow, there's this structural polysaccharide called cellulose, and cellulose is also made out of glucose, but it's bound in a very specific form that I'm sure Damon will talk to us about. And so that's been one of these big things that a lot of people have been trying to do is how can we take a structural polysaccharide, like something that's just in wood, cellulose, and turn that into glucose so that we can make a whole bunch of fuel with it in ethanol. So with that, I'm going to hand it over to Damon, but what we're really going to be talking a lot about here is enzymes. And what I used to like to say to all of my students was enzymes are the proteins that do things. There are lots of other proteins that do things, but these are the proteins that do things. They catalyze reactions. They can break them down or build things. And here's just a few enzymes that you might want to be aware of. Sucrase breaks down sucrose into its monomers. Maltase breaks down maltose into its monomers. Amylase breaks down amylose into its monomers. So cellobiase is what you're going to be hearing a lot about, is an enzyme that's going to break down, Damon?
Cellulobiose.
There you go. All right. So I'll hand it over to Damon here. And Damon, you just let me know when you want me to forward through the slides.
Okay, sounds good. So the enzyme that we're going to be looking at with this lab is cellulobiase, as John kind of hinted at, and we're going to be breaking down that substrate of cellulobiose. So cellulobiose is going to be a dimer, basically, of two glucose molecules that are bound together. And if you're breaking down a big cellulose chain, so say you're like a biomanufacturing facility that's trying to produce a biofuel from cellulose, you're going to need to break this down into these into cellulobiose, and then figure out how to separate those into individual glucose molecules that you could then take into fermentation or into another kind of enzymatic schema to produce the fuel you're looking at. So most enzymes, including cellulobiase, breaks bonds by doing positional fit. And so the cleft of the enzyme is built in such a way that when that substrate is there, it presents the bond between the two glucose molecules in a way that can be easily hit by water. And in this case, split it into the two glucose molecules. And if we think about this as a energy situation, what it's doing is by that positional fit, you're actually dropping the activation energy needed then to kind of release the two glucose molecules. And so those are kind of two different ways to think about it kind of conceptually, from the level of energy needed down to kind of a positional fit that allows that bond to be attacked by water. So if we go to the next one, we can kind of back out and think about the whole system. So this isn't our first round of trying to do basically fuel from cellulose. This was really, really hot in the late 70s. Basically, in the United States, you see a lot of investment anytime that basically oil prices spike. And then as soon as oil prices come back down, research kind of dwindles a little bit off the backside. But it's been happening enough that we've got a pretty good level of kind of tools built at this point for a potential biofuel production platform. So on the right-hand side there, what you see is kind of a very, very generalized version of what that would look like in the United States. So we would grow some sort of crop that we could then physically break down, which we're pretty good at doing. We've been doing like paper now for a couple thousand years. We've got that part down. But what we're not really good at, and we're still kind of developing the toolkit or making a robust toolkit from, is finding the enzymes now that can take the cellulose and break that down into those monomers of glucose that we can then ferment into whatever fuel is fancy at that time. And so for a while, we've been doing ethanol because humans have been producing ethanol from starches for a long period of time. But ultimately, I think what we'll see is we'll see ethanol kind of come into the market like it has been over the last 2 decades. And then we'll see other fuels start to arise. And these will come out of kind of synthetic biology pipelines where you're taking entire enzymatic pathways from one organism, moving them into another. And so you can basically feed the organism glucose, and then it can more or less poop out whatever your favorite fuel is. And so there's actually a breakthrough paper that came out just last month that I'll get to Vernier so that we can send it out in the notes. But the idea is there that they're able to now generate more or less a jet fuel for about $3.50 a gallon, which is a pretty huge change. And a lot of it comes down to figuring out these ways to break down cellulose into the glucose and then be able to ferment it up into usable fuels that have high density. So with that schematic, let's go ahead and move on to the next slide. The breaking down of this cellulose is really the hard part. Like I said, the mechanical stuff we've got down from paper pulping, but the hard part is separating the cellulose, because remember, it's in a crystalline structure because it's being used to usually protect these cells from other things in the environment, like bacteria and things like that. And so within these plants, what they need to do is figure out how to, first of all, break down the crystalline structure of the cellulose. And this a lot of times happens with heat and acid or ammonia. And then they can come in there with enzymes and then further process the cellulose until you get down to that dimer of glucose known as cellulose. And then that's where the enzyme that we're going to be evaluating today comes into play. Once you're down to that cellulose molecule, you can then send cellulose after it and break it down into that last piece of glucose. Go ahead and go to the next slide. So if we're trying to monitor this reaction, and it's really important too, because if you're trying to find enzymes that do this effectively, you need a really simple way to monitor the progress of this reaction. So on the left-hand side there, we've got cellulose. We've got 2 glucose molecules, and they're bound with a 1,4 beta linkage. Cellulose is going to positionally fit that so that bond can be attacked by water, and we can get 2 glucose molecules at the end. Unfortunately, cellulose looks the same as glucose does in water. Anybody that's ever mixed glucose and water, you know that it's clear. And so from a monitoring point of view, this is really hard to do unless you have, I don't know, like a proton NMR machine just hanging out in the back of your room. And it's also kind of hard to take those in the field. And so people in the last kind of 2 decades have tried to work out ways that we could monitor these enzymes, but not necessarily have to look at this substrate and this output. So if we go to the next slide, this is the solution that was come up for this, was to take p-nitrophenol and basically bind it to glucose in a way that that bond could be attacked by cellulose. And this now means that if I send cellulose after this substrate, it'll break it down and I'll get a molecule of glucose, clear, but I'll also get a molecule of p-nitrophenol, which if I'm in a basic solution, will be a bright yellow color. So all of a sudden, I have a really easy way to monitor, say, the kinetic reactions that I might be going after if I'm looking at new celluloses. And this is actually what we're going to use in this Bio-Rad kit, is this substrate, the p-nitrophenol bound to glucose, because it makes for a really easy system to monitor. So you can do it with a visible spec, your eyeball, or you can get analytical and pop it into one of Vernier's specs and be able to get really, really nice quantitative data about how these enzymatic reactions are progressing and what variables influence the kinetics of them.
Cool.
So
we got just one quick question. Someone was saying, if you don't mind, Damon,
yep.
like if maltose is made out of 2 glucose molecules and cellulose is made out of 2 glucose molecules, how are they actually different?
It's the bonding angle on them. So we've got a 1,4 beta linkage on cellulose. And I believe, if I remember right, I might have to look this up. I want to say maltose is alpha linkage.
I think you're right. Yeah. But that'll
question.
also be located in those resources that we put there too. Cool. Sorry, David. I didn't even
Yeah, no, that works. And that also brings up an interesting thing about cellulobases and cellulases in general. Most enzymes are super, super finicky, right, about what fits in their sites. A lot of the cellulases that we find in nature are relatively free form compared to other enzymes we work with. And that's because a lot of these are coming from organisms that are using them in the breakdown of cellulose as a food. And cellulose, right, in nature can come in a lot of different compacted forms. And so these enzymes have a little more play than some of the other enzymes we might be used to working with, say from E. coli and things like that.
Yeah, and then there's also that the 3D molecular view of amylose and other things that you'll see that amylose, which is starch, is helical and has a completely different shape than like cellulose does as well. So that's all going to be related to the type of bonding.
Mm-hmm.
Thanks for those questions, by the way. Keep them coming.
Yeah.
All right. So let's see here. Sorry about that. So there are actually two labs that exist in our book. One looks at the evolution of cellulose, and that looks at mushrooms, but there's another one, it's actually experiment number 7, that's actually also available that you can take a look at, that is in the Investigating Biology Through Inquiry. So there's 2 labs in this book that actually utilize the biofuel enzyme kit. And I can take you to the actual book there in a bit here. Let's just go here really quick to Vernier. We go to, if you search for Bio-I, that's the order code for the book. You'll go to Investigating Biology Through Inquiry. And if you look in the table of contents, there are 2 labs that utilize the biofuel enzyme kit. Lab number 7, which is the Introduction to Biofuels, Enzyme Action. And then another one that is the Evolution of Cellulose in Fungi. They both use the GoDirect Spectrovis Plus or a spectrometer, and then the biofuels enzyme kit from Bio-Rad. And if you want, we could do a live demo of that if you are all interested. It's let me just, is that all right, Damon? Does that make sense?
Yeah, that makes sense.
All right, so let me just pull the camera view over here. Hopefully you can all see this camera output.
Mm-hmm.
Great, so what I'm gonna do is I'm just gonna switch the camera really quick. Let's see here to, great, so what you should see right now is the amazing Bio-Rad kit that comes. The biofuel enzyme kit comes in a box like this, see right here. And then it comes literally with everything that you would need to do this experiment. It has a whole bunch of cuvettes that comes with. Comes with a bunch of Pasteur pipettes, a bunch of conical centrifuge tubes right here that you can see. A bunch of microcentrifuge tubes, which are really useful, that we'll show you next. And then all of the reagents come with it as well. And these really nice little packages here. So there's an extraction buffer, a resuspension buffer, a stop solution, the enzyme, and the substrate that you would use. And then also the standard. And that all comes and it has to be stored at 4 degrees C, but it's really, really simple and easy to make. I made everything this morning. It took me about an hour or so. And then you're pretty much ready to do your experiment. And what I really want to do is just show you how easy the experiment is to do itself. So I'm going to switch the camera input over here back to the other camera. And hopefully you can see this right here. Damon, let me know if I need to a little bit more light. So what I have right here is I've just got this well plate, and I'm going to do the reaction actually in these centrifuge tubes. And then I've got the cuvettes right here. And then what I've made up here is I've got like a stop solution. So the stop solution actually stops the reaction. And actually, I believe Damon makes it basic. So that's actually what turns it the color yellow.
Yep. So it's a pH 8.6. So it's not like a scary kind of stop solution. It's just a mild uptick in the pH. And it's enough to basically stop most cellular biase activity and give you that color change of p-nitrophenol.
Great. So the first thing that we do is you just take, so I've got some nice micropipettes here that I think work really well. So the first thing that you do is you take, just for the introductory activity, is we take 0.5 ml or 500 microliters of stop solution. And we put it into a cuvette. And then we take the same amount of about 0.5 ml of substrate. So here's the substrate. I just mixed it up. It's a 1.5 millimeter substrate. And we just add that, same thing, 0.5 ml, to a little centrifuge tube. There you go. And then we add 1/4 of a ml, so 250 microliters. So I've just labeled this microcentrifuge tube with yellow tape, so I know it's actually going to be 250 microliters. So then we take that of the enzyme, and I'm going to use it has you mix up two enzyme solutions, a low and a high enzyme solution. And for the preliminary activity, I'm just going to take the low enzyme solution. And then do you have a stopwatch, Damon?
Yep, I can get it going.
All right, so when I add it there, what we're going to do is just wait for 2 minutes. So I'm going to add this.
Got it.
Actually, I put it in the wrong one. I'll put it in the right one here. Here we go.
Just making sure the pipette was working. Good.
There you go.
Right,
It all looks good. Go ahead and start it for 2 minutes.
Yep.
So the reaction is going on right in here. I'll cap that.
And so one thing to be aware of too for these reagents, like that John said he just prepped this morning, these I would say are some of the most stable reagents I work with at Bio-Rad as far as the kits go. And so this is one that I have prepped weeks in advance and have just kept the reagents in the fridge and haven't had to worry about them. Bio-Rad guarantees all of the reagents for this for 1 year from when you get them. And then they usually write very conservatively about the prep on these being just a couple days in advance. But this is one that I can guarantee you can prep weeks in advance and just put it at 4 degrees. Just remember to pull them out and let them get to room temperature before using them, because enzymes at 4 degrees don't behave very fast in the classroom.
Yeah, and that's one of the things that you can use this kit to look at is what happens when you increase substrate concentration? What happens when you increase enzyme concentration? What happens if you look at increasing temperature? What happens if you look at changing pH? So that was one of the things that Damon and I wanted to show you is this is a great activity to look at inquiry and enzymes in it because it's really simple to do. The entire base reaction here takes about, this reaction is 2 minutes. And then the students will actually see a color change straight away. And then I can show you how to compare low enzyme to high enzyme, which is really simple. I would just do the same process, but then in another Eppendorf tube, I would have the high concentration of enzyme. So how are we doing there, Damon?
You're 20 seconds away from the 2-minute mark.
All right, I will get ready.
And
So
actually,
remember,
if you
go
look
ahead,
at the microcentrifuge tube that John has the reaction running in, you realize it's still very clear looking, right? And that's because even though we're having a breakdown probably of that substrate, remember it's not at a high enough pH that p-nitrophenol is getting to that resonance structure that will give you the yellow readout. And so that's why he's got that preloaded cuvette with the pH 8.6 stop buffer. So right there, go ahead and add, you're at 2 minutes.
All right, so then all that we do is we remove 0.5 ml, and then we add it right here to the stop solution. And hopefully you can see that it changed color. Yes? It's like a light yellow color. I can bring it up there if you need to, or I can shade. There we go.
Yep, that's
you can see that it's already a nice yellow color. Oh, that's a good question, Damon. How many kit reagents are for how many student teams does the entire kit work for?
So Bio-Rad designed this for 8 student stations and that they would run through a total of 6 experiments. So that's kind of the standard one that John's showing you right now, but over the course of 8 minutes where they would take samples every 1 to 2 minutes. And then looking at the changes in pH, changes in temperature, changes in enzyme concentration, changes in substrate concentration. And then the last activity is actually having the students go bioprospecting for this enzyme from the environments. And so the easiest place to see that is to go get it from mushrooms, which is really cool because the ecological niche of that mushroom usually will dictate the kinetics you see in the cellulobiase.
Yeah, which is really terrific. So just to show you, I just did that same reaction previously. Here is the low enzyme, and here's the high concentration of enzyme. And you can see, you wouldn't even have to put this in a spectrometer. Your students would be able to see that within 2 minutes, the high enzyme concentration produces a much darker yellow than the low one does. But I just did the same thing. There's instructions for how to make the high enzyme concentration. And also there's instructions on how to make, if you've got you make the high substrate concentration first, and then there's the low substrate concentration.
And what I really like about this is that you have and Vernier's done a great job of taking the kit and kind of expanding out in directions, but I think that it shows the flexibility of this lab. And so because those reagents are so stable and easy to work with, you can go really, really deep if you want to. I mean, I have people that do Michaelis-Menten derivations with this kit, all the way to people that just use this like at the middle school level, just showing enzymatic reactions taking place. And so you can kind of choose your depth. And if you need help figuring out how to do a different depth move curriculum-wise, you can always contact me around that, and I can help work that out with you.
Yeah, or if you could, the Investigating Biology Through Inquiry book that we were showing you also does that as well. It'll have really simple experiments, and then they will get more complicated as you go through. One of the things that you can do is, right, we were just looking at one reaction here at 2 minutes. One of the things that you could easily do is look at enzyme kinetics by looking at, well, what happens at 1 minute? What happens at 2 minutes? What happens at 3 minutes? What happens at 4? And the preparation would all be the same. You would have your, stop reagent. You would fill up your enzyme, basically put in your substrate and let one go for 1 minute, let one go for 2, let one go for 3, and one go for 4. And then you could actually measure those and see like what happens, which is what we're going to probably show you next. Questions on that? I mean, there's the one other thing that I really love about these centrifuge tubes is these are a great little thing that you can do to put the enzyme or the reaction at different temperatures. So by just capping up the little Eppendorf tube, you could heat this up, cool it down, etc. Temperature is a super simple one to look at. You could literally just put this in a little ice bath to cool it off. And then you could also measure the temperature, or you could put it in a nice warm bath to see what happens to the enzyme. So the kit itself is actually also really nice and tightly controlled and contained. So there's a lot that you could do with this kit. The kit is actually so useful that it's one of these things that's really great to take on the road when we're actually doing a training, because we can do lots of different things with it and it doesn't require a ton of prep. All right. Oh, turn that light off there. All right. So what about measuring? Let me go back to what about if we wanted to actually measure the absorbance or actually get a little bit more quantitative? Well, the thing that you would want to do is you would need to or would want to use is a spectrometer. And the reason for that is because that yellow color that you see there that's being reacted, its peak absorbance is down at what we call like 405 nanometers. And so to track that, you would actually really need to use a spectrometer, or what we have is a pretty affordable Godorex Spectrobus Plus. One of the cool things about our little spectrometer is you can use it over a USB port. It'll plug into a Chromebook. It'll plug into a PC or Mac. It also has Bluetooth, so you can actually use it with an Android tablet or even a phone, an iOS phone, an Apple phone, etc. It's really easy to calibrate, and the software is free. So you can get spectral analysis for free from our website. If you actually have a LabQuest, one of our little handhelds, you could use it there. And then there's also, if you have the older software that is Logger Pro, you could use that as well too. So let me just do a quick live demo of how you would do that. So I'm going to launch spectral analysis. Hopefully you can see that. And then I'll go back to visualizer here, my little, and so what I'm going to do here is I will move this over and I'll bring the spectrometer over. So this is our little spectrometer. Right here, I have a blank. I have the low enzyme, and I have the high enzyme, high concentration that is. And the low concentration of enzyme is, well, the high concentration of enzyme is twice what the low was. So I'm just going to do a really simple basic experiment. I'm just going to wind up putting these in the spectrometer and measuring them. So let's go back to spectral analysis here. So I'm going to do a new experiment. I'm going to say, I just want to look at absorbance, full spectrum. I'm going to wait for the spectrometer to warm up. Normally it would be about 80 seconds, but I've had it plugged in this whole time, so I'm going to skip it when it actually boots up here. Normally you would want to wait about 90 seconds for the lamp to warm up, but it should be plenty warm by now. So I'm just going to hit skip. I'm going to put in the blank. So if I go back over here, I'm just going to take the blank and I'm going to put it in here. Then I'll go back to the software And I'll say finish calibration. And then I'm just going to wind up putting in my low concentration of enzyme. So that was the reaction done with a low concentration of enzyme, and I'm going to hit collect. Just wait there for a minute and hit stop. And you can see that at a peak right around about 405, right around there, 400, 405, this little peak here, the absorbance is 0.63. So now I'm just going to take the high concentration. Let me move this away here because you probably don't need to see that anymore. I'll take the high concentration and put it in. And let's just see if the peak is higher. And yes, it's higher. So we can measure it quantitatively. Let's hit stop and let's measure these two. So if I look right at around 405 or 404, one's at 1.5 and one's at 0.6. So it's a little bit higher than twice as much, but that's pretty good for me mixing everything up non-quantitatively, you know, eyeing everything with a graduated cylinder and everything. I would say that's pretty good. So this is just a really simple way that you could actually measure the absorbance of these two reactions. There is a way actually also in the book where you can create a standard curve, and then you can actually calculate the real amount of product that's being produced. But one of the things that Damon and I would like to remind you is you don't have to do that. You can look at lots of trends without having to use the standard curve. There's lots of different things that you can look at. If your basic idea is just to show students how things kind of happen, you don't need to do that. What was the blank? So the blank is interesting. It's really just stop solution with distilled water. And really what you're trying to do is you want to put a little bit of distilled water in, then also with that stop solution, which is that buffer. And so that's what the blank is. But it tells you what the blank is actually in the lab instructions as well. But yeah, that's what the blank is. Other questions?
I was gonna say, like, one of the things I just want to highlight, because this is why one of the things I really love about using Vernier stuff on this is that I don't have to give the students what wavelength to look at because they can find it. And what I like to do is have them think about what they're seeing visually first, and then make a hypothesis about where it's going to show up on the spectrum. And they start to actually get an intuitive feel for this after you've done a couple of labs using this software for it, which I think is a really unique perspective for them. Instead of just having, oh, here's the number to punch in there, and they don't really conceptualize what that means. So anytime you can kind of get them to start to get an intuitive feel for where this is going to show up in the visible spectrum is wonderful. And I really like that this display helps them do that.
Yeah, I think it's really great, especially for, I would say, biology students. Like if you were to look at chlorophyll with our spectrometer, what you would see right away is no absorbance in the green, but absorbance in the blue and then absorbance over in the red. So it's a great way for students to understand that what a solution absorbs is not what they're actually, the color that they're actually seeing. All right, so what I wanted to do next is I actually have a ton of sample data that we can give you if you wanted to take a look at. So when we did this actual activity, I went through and did a whole bunch of different experiments, and I can actually show or share with you some of this sample data or give it to you even if you're interested. So one of the things that I was going to do here is move over, and I think I was going to let me close Spectral. And then I can open up. Let's see, here is, let's take like a closer look. Let's see, but a closer look at temp. So this is actually data that I actually calculated previously in LoggerPro, and I just entered it into graphical analysis manually. And what you can see right here is these are these different reaction rates that we were talking to you about. So like I did the reaction for 1 minute, 2 minutes, 3 minutes, 4 minutes, and then 6 minutes. And what you can see right away, I'm just going to apply a curve fit here. What you can quickly see is that when the reaction was done at 34 degrees C, it has the highest rate. The slope is the tallest, or the steepest. And then at 22 degrees C, that's this blue one right here, that's intermediate. And then as it gets colder, that rate just decreases and decreases. And once again here, we're just looking at time on one axis and then the absorbance. We're not actually looking at the true amount of paranitrophenol that's been created, although there is a way to do that I could show you as well.
And one thing to be aware of too with just this enzyme is that where we're getting it from currently is, I think, almond husk. And this enzyme is really, really thermally stable. And so if you want to actually denature it, right, and show failure, I'm an industry person. I love to see failure. Students hate to see failure, but it's really important to know where failure is. You need to drive this up to over 85 degrees centigrade to see failure due to denaturation.
Oh, wow, that's really interesting. So you'd have to get that high before it begins to denature.
Yeah,
that's really interesting.
And some of the places where you might derive cellulose from will get you past boiling. So I was doing this with some students, and we used Schizophyllum commune, a very common mushroom you can find in most habitats. We had to use salt solutions in order to be able to heat it up high enough that we could get it to denature. So we got up to 125 C before it actually denatured the protein.
Wow. That's pretty amazing. So I can open up another one for you here. Let's save that. Choose a file here. Oh, let's see here. Hold on here. I'll open up another file.
And anytime you're looking at these different variables, I think it's really important to have students think about why might those be the endpoints of where this enzyme survives. And for a lot of these cellular biases, we're getting them from fungi. And so they happen to live a lot of times in very acidic environments and environments that heat up during decomposition. And so that's why you see these abilities to handle pretty high temperatures compared to other enzymes we're used to working with that denature at, say, 65 C and things like that. And then also most of these cellular biases will perform really well at low pHs, but then fall off really quickly once you start to make your way into slightly basic conditions. And that's why that stop solution in the kit doesn't have to be a very strong base. Like pH 8.6 knocks out pretty much every cellular bias we've looked at.
Great. So yeah, so that's why in this graph that hopefully you're seeing right here, you could see that we did this reaction over a period of time up to 8 minutes. And you could see that at pH 8, there's almost no reaction. pH 6.3, it's intermediate. And then at pH 5, it's much steeper. All right. So other questions? Oh, did you mention this graphical? Oh, that's a good point. So I'm using two different types of software here. I'm using spectral analysis, which is free. And then I'm also using graphical analysis. There is a free version of graphical analysis that you can get that you can just do manual data entry into. And then there is a pro version. Like if I wanted to share this data with you, I could send it over the internet and you could get it. And that you would need to then use graphical analysis pro. And there is a way to do that. But if you just wanted to get either versions of these software, let me go back to Vernier. If you go to the top corner here under downloads, you'll see that you can get graphical analysis or spectral analysis, and you can download them directly. Spectral analysis is free. If you go to Vernier graphical analysis, you'll be able to download it now. There's a Windows version, Mac OS, Chrome, iOS, Android, etc. It should recognize what platform you're on, and you should be able to do that. I'm just using graphical because I had data that I had in Logger Pro or another application. I've done all these calculations, and then I just manually put it into graphical analysis, almost like Excel. I just like to use graphical to do stuff like that because it's really easy to do fits and to make graphs. Other questions? Like we said, we went over all the ideas for inquiry, other things that you could do. Just remember this, the lab that we've been talking about right here is the Introduction to Biofuels Enzyme Action. There's two that are in the back of the book. Another one that looks at mushrooms. Damon and I have done an entire other webinar on that. So you can actually go to the website, the Vernier website, and search for mushrooms, biofuels, and you'll come to a completely different webinar where we actually show you how to grind up mushrooms and do this sort of activity as well. That's actually really popular. And then if there's other labs or other things that you're interested in, please let us know. One of the added activities that's actually in that I did using this kit is I took the Cellulose enzyme and I bought some Cellulose. There is no Cellulose in the kit, but you can buy that. And then I just looked at, hey, can yeast metabolize, Cellulose? Like how well can they do that? They can't metabolize it very well at all. They don't produce a lot of CO2. But if you throw in a little bit of Cellulose into that Cellulose and mix it up and feed it to them, it's a lot like sugar. So you can see that the CO2 gas sensor will produce a lot of CO2 gas with yeast that are fed the enzyme and the substrate there. If you already have one of our books, like if you already have the Investigating Biology with Inquiry book, I just wanted to let you know that you can get the contents of all of this and the updated instructions for free. You just want to go to our website in the top right-hand corner, go to where it says log in or register, and create an account. And if you create an account, make sure you use your email, your school email, and then it will automatically populate or go out and find, hey, if there's a book that was bought that is being used by a teacher at the school that you're at, you will actually also wind up getting access to it too. So do that first before you go out and buy the book. You might already have access to it. Like I was just saying, like there's lots of personalized training. So if you wanted me or someone else on my team to do like a webinar with you, like it's just a Zoom, not really a webinar like this, but just a Zoom, we would definitely do one of those for free as long as it's under 60 minutes and it could be on anything. But we could easily show you how to use this kit, help you out, etc. Never be afraid to get in touch. I know that Damon probably also has other information that he can give you on how to contact him. But this webinar, you can also will be able to find it on our website if you look over at webinars under educator training, and there's lots of other great webinars that are there. And we even have a YouTube channel which has lots of information and great stuff on it. So other questions? No questions on the Bio-Rad kit. Oh, I should show them. I should show you them all that. Go ahead, Damon. Do you want to talk a little bit? I'm going to show them the little our little website where we have our nice little agreement with Bio-Rad and.
Yep. I'll drop my email in the chat. So if somebody has a direct question about the kit that comes to them later, you can just email me. I was going to say, John, if you ever want to revisit your yeast experiments with the Cellulose, don't use yeast. Use koji. Use aspergillus. Because you need a fungus that actually produces cellulose into the environment to break down cellulose, where yeast is used to taking in sucrose.
Gotcha.
And so koji would be a better fit for that. And then you could use the Vernier probe set to monitor the CO2, which would be really, really cool. This lab has been out for about 9 years now. And so it's been run through the gamut of everything you could possibly imagine done to it. So if there is a way that you want to try and take this and go some other direction with it, feel free to reach out to me. And I've probably seen that direction be at least tested. And I can tell you, yep, that's a good idea. Or here's the asterisks about running in that direction. Because it's one of the things I really love about working for Bio-Rad and their kits is they're built with such quality that it means you can do a lot of the things that they didn't intend you to do with them, which means you can kind of tweak things to get your specific learning objectives out of them. So if you ever need that type of help with the biofuel kit or any other Bio-Rad labs, feel free to reach out to me. That's one of the primary things I end up doing in this role is kind of tweaking things to get what folks want out of them.
I completely agree. We've been working with Bio-Rad for a long time, and every time we get one of their kits, it works. We never have any problems with it. There's just terrific customer service at Bio-Rad, and we really appreciate that. If you ever want to get in touch with me or someone in the biology department, all you have to do is email biology@vernier.com and just tell us what you need, and that'll go to one of the people in the biology team, and I'll probably see those or two other of my colleagues. But that's probably the best way to get in touch with us. We do have this really cool spot on our website that highlights our relationship. It's the Bio-Rad Laboratories and Vernier Software Technology webpage. It has an overview of how we collaborate, work together. And then it shows a whole bunch of Vernier Bio-Rad kits. And what's really neat is it'll tell you like what is the experiments that you can use, what is the product. This little link will actually take you out to the Bio-Rad website, so that you can actually like take a look at it and order it there. That's one of the very few cases on our website where we tell people to go somewhere else, but we love Bio-Rad so much we do that. And then the other thing that I wanted to show you really quick is you can get the actual instructions for these labs just by clicking on these little links. So if you click here on Introduction to Biofuels Enzyme Action, that just downloaded that for you right there. And that's going to have the teacher's instructions and also the student instructions as well. So if you want to get access to these labs without having to buy the entire book just to play around with them and use them, they're all located right here. And all that you would need to do is search for Vernier and Bio-Rad up at the top to find that. Oh yeah, no, someone just, yeah, any wood rotting fungi would work great for the solid bias prospecting. That's one of my favorite things to do in this lab is to compare, say, like shiitake to generalist mushrooms, to a endosymbiotic mushroom like chanterelles or something like that, and just to see that they're very different. Mycorrhizal fungi would be very, very different to those that are specialists on rotting wood.
And then even in the in-between space, if you're looking at ones that work on kind of pre-digested things like cow dung, so if you're looking at your button mushrooms, they'll have intermediate kinetics compared to a strong wood rotter like shiitake versus a mycorrhizal, which has usually almost zero activity.
Almost zero. But like I said, that you can actually find that webinar also on our website, where Damon and I have gone through that as well. You'll be able to find that there as well. So thank you so much. I don't think there are any other questions. If there's anything we can help with, please let us know.
Awesome. Thanks, everybody.